and welcome to episode 12. This is the final episode in the series introducing the cell. In the last few episodes, we've explored quite a bit into all the little detail of the cell. We've discussed the role of proteins and enzymes in cellular functions. We've talked about how cells talk to one another, the lipid bilayer membrane, the endomembrane vesicular system, the organelles, and we've even talked a little bit about the basic metabolic and respiratory pathways. In this episode, I'm going to discuss the 4 stages of the cell cycle. That is, the stages a cell naturally goes through before it replicates itself, creating 2 new daughter cells. Recall the cell theory that I mentioned in some of the earlier episodes. One of the original tenets of the cell theory was that cells arise from pre-existing cells. This raises an obvious question. How does a cell arise from another cell? Or conversely, how does a cell give rise to daughter cells? This process, as you might have surmised, is called cell division. To really get to the answer of our question, we have to look at how cell division works on a molecular level. There are 2 types of cell division, with 2 very similar but fundamentally different chemical processes. The first type is called mitosis. During mitosis, a cell will copy its organelles and chromosomes, and split them evenly between the 2 emergent daughter cells. Because each daughter cell shares a copy of the parent cell's genome, the daughter cells are clones of the parent cell. As the parent cell itself is a clone of its parent cell, you quickly realize that all the cells of a single organism's body are all clones of the same genome. The single individual organism has a body composed entirely of cells, which grew and replicated from an original embryo. In asexual species, the offspring is a clone of the parent. But in sexual species, the offspring is not a clone of the parent. Instead, there's genetic variation between the offspring and the parent. So where does this genetic variation come from? The answer brings us to the second type of cell division, which is called meiosis. During meiosis, a cell doesn't copy its chromosomes, but continues to split them up evenly between 2 daughter cells, which are called gametes. In this way, each daughter cell only has 1 copy of the parent cell's genome. Humans, and many other sexually reproducing species, are diploids. This means that each of our cells have 2 copies of our chromosomes. Triploid cells possess 3 copies of their chromosomes. Haploid cells only possess 1 copy of their chromosomes. So when our diploid parent cell undergoes meiosis, it produces haploid gametes. These haploid gametes each have a single copy of the full chromosome. Because the vast bulk of cells in our body are somatic cells, or body cells, they only perform mitosis and only produce clone daughter cells. Tissue differentiation begins with stem cells, although stem cells are a whole episode in and of themselves. To be very brief, stem cells basically code their daughter cells to express only a certain collection of specific genes. If you listen to the previous episode about genetic expression, you should understand that different types of cells, even though they have the same total genome, only express a particular specific subset of those genes. And it's through this specific expression of only a small set of genes that the cell is able to differentiate itself into a neuron, or a muscle cell, or a retinal cell in your eye, or something like that. Your nerve cells and skin cells, for example, both contain the exact same quantity of genetic material and the exact same genome. However, these cells each express a different collection of genes within the shared genome, which causes them to develop into different shapes and perform different functions. The gonadal structure, the testicles in males or the ovaries in females, are organs that produce gametes through meiosis. Sperm cells and eggs are both gametes, and they possess only 1 copy of their genome. When an organism has diploid cells, the cells can express traits from either copy of the genome. That is, the genome codes for specific traits, but there can be different versions of this trait called an allele. Eye color, for example, is a trait. Everyone has genes that code for eye color. But we all have different variations on that gene. We all have eye color alleles. Most people have alleles that code for a shade of brown, like light brown, dark brown, but other people have alleles for green, blue, or hazel-colored irises. The haploid gamete only has 1 copy of the genome, so it has no choice between alleles like the diploid cell. However, this is really important for sexual reproduction. The sperm gamete fuses with an egg gamete, and each provides its copy of the genome to a single daughter cell, a fertilized egg. Each copy, one of the father's genome and one of the mother's genome, then combine and exchange information within this new fertilized egg, and this becomes the new genome of a new individual. All the cells that arise from this fertilized egg share the new genome. This clump of rapidly dividing cells will become the offspring. For example, the person with alleles for green eyes. So let's get back to the cell cycle. There are 4 stages to this cycle. In order, there are the first gap stage called G1, S stage, a second gap stage called G2, and M stage. M stands for mitosis or meiosis. Each one takes place in the same relative position in the cell cycle. The G1, S, and G2 stages are all a part of interphase, which is the stage of any cell that's not currently dividing, or an M stage. So basically, a cell is either in interphase or M phase. If it's in interphase, it's either in G1, S, or G2, and it's just growing and functioning like normal. It's doing all the normal things a cell normally does. But then it leaves interphase and comes to M phase. And M phase, for mitosis or meiosis, is where these processes actually occur. And so this is the stage of the cell cycle where the cell divides. Both the G1 and G2 stages are characterized by this mundane normalcy. During both gap stages, the cell is just doing its thing, respiring oxygen and carbon dioxide, communicating with proximal cells, doing general upkeep and maintenance on its internal structures, repairing and cleaning up its DNA, that kind of thing. At the end of G1 stage, there's a checkpoint. And this is really important. This checkpoint is like a self-check test in a single-celled organism, and a community evaluation of sorts in multicellular organisms. If the cell passes this check, it continues on with the cell cycle to enter S phase. However, if the cell fails, it enters a dormant stage called G0, which I'll discuss more in a moment. The G1 checkpoint is like a full-body diagnostic. The quality and the health of the cell as a whole is taken into consideration. The cell must have undamaged DNA. It must have the proper size to be able to produce a viable daughter cell. It must be in some kind of environment that isn't nutrient stressed. The cell must be able to recognize and respond to chemical signals from other cells, called social signals. If any of these measures aren't up to par, some mechanisms within the cell will act to immediately halt its progress in the cell cycle. If the DNA is damaged, for example, a protein called P53 activates specific genes in an emergency response. It's like pulling the fire alarm. So these activated genes will either halt the cell cycle until the damage can be fixed, or it'll choose to just abandon ship. This latter option is like totaling your car. It's a total loss, pretty much impossible to fix or replace without risk or egregious expense. If the DNA is sufficiently damaged, the P53 protein can trigger apoptosis, which leads to the voluntary death or the suicide of the cell. The G2 phase has a similar checkpoint, but it focuses on integrity of the genome. If the cell's DNA is damaged or degraded in a significant way, the cell will not be allowed to enter M phase. Recall the G0 phase I mentioned briefly. This stage is basically an arrested G1 phase. The cell is no longer in the cell cycle. It doesn't bother with checkpoints anymore. It doesn't synthesize a copy of its DNA, and it doesn't replicate. The G0 phase can be described as post-mitotic, as it no longer experiences mitosis. Many adult cells naturally enter a non-reproducing G0 stage once they mature. Some examples of these cells include nerve cells, which almost never reproduce but can last for your entire life, and muscle cells, which are too long and protein-packed to undergo a normal form of replication. The G1 checkpoint is heavily dependent on the social signals the cell receives from other nearby cells. This communal signaling ensures that all the cells in a tissue or local community are going through the cell cycle in a kind of rough synchronization. This synchronization isn't perfect, but instead it induces a kind of cyclical pattern of growth and division in the cell community. First, various proteins and chemicals that enable proper growth are released by all the cells, or at least by the cells in a similar stage in the cell cycle. These growth factors begin working by initiating the production of other regulatory proteins, namely E2F and G1 cyclins. Let me define a cyclin real quick. Cyclins are small protein compounds whose concentration cycles between high and low within the body of the cell. As the cell undergoes some kind of behavior, the concentration of a cyclin will rise. As the behavior changes or comes to an end, or is otherwise modified, the concentration of cyclin decreases. As I explained in a previous episode with a calmodulin example, the concentration of a specific chemical can be the trigger that induces a response. Anyways, the cyclin binds to a kinase, which is a type of protein that can catalyze the phosphorylation of another protein, so it's kind of like an activator. These kinases are only active when it's bound to a cyclin protein, so its activity is dependent on the concentration of cyclin in the solution. As a reasonable course of events, this kinase is called a CDK, or a cyclin-dependent kinase. During the G1 checkpoint, the concentration of CDK cyclin, or cyclin-dependent kinase, the kinase molecule, bound into a complex with a molecule of cyclin. And so this forms a CDK cyclin complex. During the G1 checkpoint, the concentration of the CDK cyclin complex increases as the growth factors continue to induce the production of cyclins. The more cyclins you have, the more they bind to CDK, the more CDK cyclin complexes you have. It's not terribly complicated. The other protein produced by the growth factor, E2F, is bound to and inactivated by a protein that suppresses tumor growth, called RB. RB also inactivates the CDK cyclin complex. In time, the CDK cyclin complex is dephosphorylated, and this activates it. The activated CDK cyclin complex then catalyzes the phosphorylation of more RB. The addition of this large, negatively charged phosphate group induces a major conformational change in the RB protein. This conformational change makes RB unable to hold onto E2F, and so E2F is released into the solution. So the E2F molecule is no longer inhibited, and this allows it to activate specific genes and induce the cell to enter S phase. So basically you have a protein called E2F, and E2F can trigger genes to make the cell go into S phase. But in order for E2F to work, it has to get out of the grip of a protein called RB. And so RB just holds on tightly to this E2F and doesn't let it do anything. And as a result, the cell doesn't go into S phase. And so these CDK cyclin complexes increase in concentration, cyclically in the cell. The CDK cyclin complex is also inactivated, but it loses a phosphate group, and this makes its conformation change, and it becomes activated. So when the CDK cyclin complex is active, it can take that phosphate group that was just released a moment ago and phosphorylate the RB protein, so it can stick that group onto the RB protein. And now the RB protein then experiences a conformational shift, and this conformational shift weakens its grip on E2F, and this releases E2F, and E2F gets to work. When the cell enters S phase, it's still in interphase. It's still doing all the things that it normally does, but with one exception. The S in S phase stands for synthesis, as the cell begins to synthesize DNA. It's not that the cell is making DNA from scratch. Instead, it's copying its DNA using its original DNA molecule as a template. Each copy of the genome takes the condensed form of 23 chromatids, protein-dotted clumps of tightly wrapped up DNA. I discussed this in detail in the previous episode. The original chromatid and the copy chromatid are called sister chromatids. Copying the entire genome is a massive undertaking for the cell. It requires a titanic amount of nucleotides, not to mention ribose sugar and phosphate groups for the backbone. Creating copies of all the organelles requires a similarly titanic amount of phospholipids and the associated membrane proteins. All of these resources take considerable energy to organize and piece together, and this means that S phase is characterized by increased metabolic activity to accommodate the synthesis of a whole new copy of the genome and a copy of all of the cell's internal structures. If the cell passes all the interphase checkpoints, it enters M phase. It finally enters M phase and begins mitosis or meiosis. Here, each sister chromatid pair is split up, and each independent daughter chromatid in the pair goes to one or the other daughter cell. Each daughter cell thus has one copy of the same genome, emerging from M stage to find itself right back in G1 phase at the start of the cell cycle. This might sound straightforward, but M phase is anything but simple. There are 5 stages within M phase, corresponding to the steps of mitotic cell division. These 5 stages of mitosis are, in chronological order, prophase, prometaphase, metaphase, anaphase, and telophase. Prophase is the beginning of M phase, marked by 2 major events. The first event involves the genetic material of the cell. The strings of DNA condense into compact, protein-studded structures called chromosomes. As the DNA was replicated in the S phase, the DNA in M phase condenses into a pair of chromosomes. The second event involves the beginning of the formation of the spindle apparatus. This involves two structures called centrosomes, which each have two identical subunits called centrioles. From the centrioles, microtubules extend into the cell to organize and structure the subsequent steps of cellular division. Because each chromosome goes to one end of the cell, their microtubule fingers reach across the cell, from the ends to the middle. Some of these microtubules will begin to overlap with each other as they extend, and these ones are called polar microtubules. So once the chromosomes have condensed and the spindle apparatus is formed, the cell enters the next stage of M phase, called Prometaphase. In Prometaphase, the nuclear envelope dissolves and disappears. This exposes the condensed chromosomes to the rest of the cell, which is critically important for the next step of Prometaphase. The microtubules that were set up in prophase now extend throughout the cell, connecting to structures at the center of each chromosome called a kinetochore. If you imagine the traditional X-shaped chromosome, the kinetochore is the globular structure at the middle of the X that helps hold everything together. Some of these microtubules bind to these kinetochores, which enable the next stage of M phase, called metaphase. In metaphase, the microtubules align the chromosomes into a flat plane running down the center of the cell. This mass of chromosome pairs, organized into the plane at the center of the cell, is called the Metaphase Plate. The formation of the Metaphase Plate marks the final step in the formation of the spindle apparatus. The polar microtubules have overlapped enough to reach the opposite centrosome on the other side of the cell. The microtubules attached to the kinetochores are stabilized by a network of thin astral microtubules, which form a web-like lattice between the kinetochore microtubules and the inner surface of the cell's plasma membrane. Because of the dynamic nature of the structure of microtubules, they decay at one end and grow at another. The kinetochore microtubules decay and shrink at the centrosome end, called the negative end, and they slowly grow at the kinetochore end, or the positive end. This induces directionality in the microtubule. Imagine a microtubule of static length. On its left end, the subunits decay and break off easily. On its right end, subunits are added more frequently than they're broken off, and so the microtubule grows. If both ends of the microtubule stay in the same place, but the subunits continue to decay off the negative end and get added to the positive end, then the individual subunits will slowly move down the length of the microtubule, from the right-hand side to the left-hand side. Alternatively, if both ends are decaying, the subunits will be relatively motionless, but anything that can hang on to a dissolving end of the microtubule will be carried along as the tubule shrinks. This quality is important for understanding the next stage of M phase, called Anaphase, and it begins when the bond between the sister chromatids is broken as each daughter chromatid is pulled by the centrosomes to either end of the dividing cell. In this way, each copy of the genome is pulled to one end of the dividing cell. The daughter chromatids are pulled to the centrosomes by two different mechanisms. The first mechanism involves kinetochore proteins, which collar the positive end of the microtubule. And so as the microtubule shrinks, these subunits are gradually brought closer to the centrosome, dragging the chromatid along with it. The kinetochore on the sister chromatids has proteins that form a collar-like structure, so this collar wraps around the positive end of the kinetochore microtubule. And as the positive end of the microtubule frays, subunits, monomers, spill out into the solution, and it widens. It frays, kind of like a rope, with a loose end. This widening pushes against the collar, and it pushes it further down the microtubule that hasn't begun to decay yet. In this way, the kinetochore hangs on to the dissolving end of the microtubule, and it gets carried along with it as the microtubule dissolves. This event is also an integral part of the first M phase checkpoint, which makes sure that the sister chromatids separate properly. If the microtubules don't connect to the kinetochores, the daughter cells will have an inappropriate number of chromosomes, which can lead to serious developmental problems, if not outright death. The second mechanism involves the centrosomes themselves, which are pushed by polar microtubules further into opposite corners of the cell. These two mechanisms force the sister chromatids apart, with one copy going to each corner of the cell. Anaphase comes to an end when one copy of each chromatid is dragged to a centrosome. This basically means that anaphase ends when two clumps of cloned chromatids are clustered in each corner of the cell. Once all the DNA has been copied, and each copy pulled into the opposite ends of the cell can undergo the last stage of M phase, called telophase. Recall the nuclear envelope that dissolved in Prometaphase. During telophase, this nuclear envelope reforms around each clump of cloned chromatids, and this forms two new nucleuses. Within these reformed nuclear envelopes, the condensed DNA in all of the chromatids begins to loosen and unravel. By loosening and unraveling, the DNA makes itself far more accessible to the protein machinery that reads, translates, and transcribes the information in DNA into proteins. I covered this in the previous episode about gene expression. So outside of the nuclear envelope, the structural fibrils on the inner surface of the cell membrane begin to pinch off in the middle. These structural fibers are actin filaments, and they form a ring around the middle of the membrane, kind of like a belt or an outline of the Metaphase Plate on the inside of the plasma membrane. Myosin protein fibers attach to the actin, and they begin to pull themselves along, tightening the actin belt. This is the same mechanism through which muscle fiber works, but that's a different discussion for a different episode. Anyway, as these actin fibers are connected to the inner surface of the plasma membrane, the tightening pulls the membrane with it, creating a deep crevice called a cleavage furrow. The cleavage furrow deepens as the actin belt is tightened. The actin belt eventually closes off, it tightens completely, and this causes the cell cytoplasm to split into two separate bubbles, each containing a nuclear envelope of its own, with a full copy of the genome and a full complement of the copied organelles. The cytoplasm is pinched off, the membrane separates, and two new daughter cells are born, each possessing the exact same genetic information as their parent cell. This act of pinching off the cytoplasm and the separation of the membrane into two smaller daughter cells is called cytokinesis. There's a substance called M-phase promoting factor, or MPF, which induces a cell in interphase to enter M phase. To understand how MPF works, we have to understand the chemical structure of MPF and how it interacts with other molecules. So the physical structure of MPF is defined by two subunits, belonging to chemical groups called cyclins and kinases. Together, these two subunits make an MPF molecule, or an MPF complex. Cyclins are molecules whose concentration in a cell cycles from high to low. In the case of calmodulin, higher concentrations in an epithelial cell of your blood vessel causes the blood vessel to expand. In the case of MPF cyclin, the concentration rises during interphase to peak in M phase, where the concentration then crashes back to zero, which ends M phase. The concentration of cyclins then increases again during interphase, only to peak and crash again during the next M phase. The second subunit is a kinase, an enzyme that can catalyze the phosphorylation of another protein. As the concentration of cyclin increases during interphase, it binds with the CDK. Two phosphorylation reactions are used as regulatory tags to keep the CDK cyclin complex inactive until enough of it has been produced to effectively initiate M phase. At the end of the G2 phase, immediately before M phase, an enzyme is produced that dephosphorylates the MPF complex. This activates it and allows the kinase subunit to catalyze phosphorylation reactions. After M phase has been initiated, the concentration of MPF crashes. So what happens to the CDK cyclin complex to cause its rapid destruction? The MPF is regulated by negative feedback. As MPF is necessary to enter M phase, it follows that M phase cannot happen without high concentrations of MPF. Within M phase, an enzyme is activated. This activated enzyme begins to deactivate all the MPF that it can find. Another regulatory process works through targeted destruction. During anaphase, or the second to last step in M phase, an enzyme complex is activated, and this begins to hunt down and tag the MPF cyclin. This enzyme complex tags the molecules of MPF cyclin with small proteins called ubiquitins. The ubiquitin tag marks the protein for destruction at the hands of another complex, called the proteasome. Through these two mechanisms, the concentration of MPF is self-regulating. The concentration is raised gradually through interphase, and then activated at the end of G2 phase in high concentrations to initiate M phase. Various proteins are activated in M phase, which then work to deactivate and digest the subunit proteins in MPF. This negative feedback drastically reduces the concentration of MPF, ending another evolution in the cell cycle and putting the cell back at the start of interphase. The completion of cytokinesis indicates a successful passage through the second M phase checkpoint. If the chromosomes don't separate properly, the proteins that degrade the cyclin molecules aren't activated. If these proteins aren't activated, cyclin molecules aren't tagged with ubiquitin, and they aren't degraded. As a result, the concentration of CDK cyclin complexes, or MPF, doesn't drop. The concentration instead stays high. If the concentration of MPF is high, the process of cytokinesis is stalled. The proper handling of protein complexes are also involved in the regulation of the G1 checkpoint. If the RB protein is defective, it won't be able to bind and deactivate the E2F protein. Without regulation, the E2F protein will continually push the cell into S phase. Cyclins can also be overproduced by the presence of excessive growth factor, or some issue in the signaling pathway that can't shut down cyclin production even without growth factor. Cyclins can also be overproduced by the presence of excessive growth factor, or some issue in the signaling pathway that can't shut down cyclin production even without growth factor. Problems with either RB or cyclin production can cause cells to override the G1 checkpoint and move into the S phase prematurely. It's important to understand the healthy and proper functioning of the cell cycle, because errors or mistakes in the cell cycle can have disastrous consequences. Uncontrolled cellular replication creates masses of quickly reproducing cells called tumors, or cancers. Cancer is basically a group of cells that have, through DNA mutation or some kind of interference, lost the ability to regulate their own reproduction. Cancerous cells have two specific problems that prevent them from regulating their reproduction. First of all, these cells can't turn off the proteins involved in growth and reproduction. These proteins are active, but for whatever reason, the cell just can't inactivate them. Second, cancer cells cannot express genes that work to suppress cancer by shutting down the cell cycle and damage cells. So even if the cell recognizes a problem, it can't shut down the factory. It can't stop the assembly line. The cell can't sound the alarm or call in the cavalry. The cell has no mouth, and it must scream. If a cell suffers from these two problems, it'll become cancerous. As almost all cells need to reproduce, almost all tissues are susceptible to developing cancer. Skin cancer can be one of many different kinds of tumors that form from damaged skin cells. Rates of skin cancer are higher in areas with more sun exposure, as the UV radiation induces mutations in the cells on the outer layers of your skin. These mutations can disable your cell's ability to regulate reproduction, and before you know it, you have a tumor. Lung cancers can be caused by exposure to airborne chemicals or particulates, like the smoke from cigarettes or the metal fumes in a welder's shop. These carcinogens aggravate and damage the delicate cells of the alveoli tissue in the lungs, encouraging them to become cancerous. In breast cancer, tumors form from the cancerous cells in mammary tissue. Cancerous cells can develop into tumors in the colon, the bladder, the thyroid, the testicles, and the pancreas. Cancer in the retina, the layer of light-detecting cells in the eye, can lead to a retinoblastoma. Cancer in the blood marrow can lead to leukemia, characterized by the production of impotent, inefficient, or malformed white blood cells. The point is, cancers can form almost anywhere. They can be triggered by a huge variety of environmental or genealogical factors. There are generally two types of tumors: malignant tumors and benign tumors. Benign tumors are usually harmless. The cancer cells will continue to reproduce, and the tumor will continue to grow, but the tumor itself is more or less encapsulated. It won't spread, and this means that the tumor exists as a single mass, and this mass can easily be removed with surgery. Sometimes not so easily, but the fact still remains that as a single mass, it's much easier to locate it and take it out. While benign tumors are generally non-lethal, they aren't totally harmless. The tumor can be positioned somewhere such that its growth applies pressure onto a softer organ. Benign brain tumors, for example, can cause serious problems by pressing hard against the adjacent brain tissue. If they keep growing, the pressure increases. Benign tumors in the throat can impair speech, respiration, and comfortable head movement. The fact that benign tumors don't spread is what separates them from malignant tumors. Malignant tumors are dangerous. They aren't encapsulated in any sense, and so they can spread throughout your body. Individual cells, or clumps of cells, can flake off from a malignant tumor, entering the bloodstream or a lymph vessel to be rapidly spread throughout the body in a process called metastasis. These flakes of cancerous cells can get lodged or be deposited anywhere else in your body, anywhere your vasculature takes it, and wherever they lodge, they'll grow into new tumors. The malignant tumor spreads by basically dumping little cancer seeds into your bloodstream. These cancer seeds then lodge in all of your tissues, where they absorb nutrients from healthy cells and they grow into new tumors. These new tumors produce little flaky cancer seeds of their own, and before you know it, the cancer has spread again. Catching a malignant tumor early is very important, as it can be killed, removed, or contained before it spreads. If you aren't aware of a malignant tumor for a long time period, by the time it gets diagnosed, it may have metastasized. It may have spread throughout your body such that there's too much cancer to remove surgically, without throwing you through a metaphorical cheese grater. The reason we use chemotherapy is because it's an indiscriminate treatment. It kills pretty much all cells, but hopefully it kills more cancer cells than it kills healthy cells. When you use surgery, you're specifically going in to take out that one little tumor. But if you have a malignant cancer and you have little tumors all over your body, if you want to get those out with surgery, you're going to have to cut up your body savagely. You'll have to make incisions in many, many different locations, all going to varying depths, in the tissue to get the tumors. So that's why we can't really use surgery to treat malignant tumors, because you basically end up just cutting the person up. And recovery from that kind of surgery would be a nightmare. Without pain medication, it would be horrible. And even on pain medication, it would still be, exceedingly painful and uncomfortable. You've basically had chunks of your body all over taken out. That's an extremely traumatic experience for your tissue. Alright, I went on a rant. So to wrap things up, I should say that the cell cycle is extremely important to pretty much every living organism. The cell cycle and its series of carefully maintained checkpoints prevents the cell from reproducing if it has genetic damage that can be passed on to its daughter cells. The tumor suppressor genes, the checkpoints, and apoptosis are all tools the body uses to prevent errors in the cell cycle from running amok and creating tumors. Naturally, this system isn't perfect, as people still get tumors, but it works remarkably well. If we didn't have it, we'd be covered in tumors and would probably die from cancers at a very young age. But because of the cell cycle, our bodies can grow and heal in a relatively controlled context. In a future episode, I'll cover meiosis. I'll discuss how meiosis uses different mechanisms than mitosis to separate the genetic material into haploid daughter gametes, and how this genetic material interacts during fertilization to create a new diploid genome. But that's it for this episode. I hope I was able to explain the cell cycle in a way that you can easily understand, and I hope you learned something cool. I know it was a little complicated though, and especially during the middle of the cell cycle, things got a little intense. So this episode wraps up the mini-series introducing the cell, so hopefully you can feel confident in your knowledge of the basics of cellular physiology and activity. Thanks for listening. If you enjoyed this episode, hit the like button and subscribe. Share the episode with a teacher, a friend, or a classmate. If you want to support the show, follow us on social media and consider buying something from the official store. Follow us on Twitter. Our username is @BioLogicPodcast. To buy something from the store, go to redbubble.com and search for BioLogic, or go directly to the store at redbubble.com/people/biologic/shop. Become an official patron of the show. Sign up at patreon.com/biologicpodcast. Your support really helps the show, so if you like it, send us some love. And as always, thanks for listening.